Hello, everyone, and welcome to this Around the World Australia webinar, which is hosted by the Australasian Gene and Cell Therapy Society in conjunction with the Coalition of Gene Therapy Societies. My name is Samantha Jin, and I'll be moderating this webinar together with Dr. Livia Carvalho. There'll be a short Q&A session after each speaker, so please type your questions into the Q&A function throughout the presentation. And it's my pleasure to introduce our first speaker, Dr. Alexandra O'Donoghue. Alexandra is a postdoctoral researcher in the Bioengineering and Molecular Medicine Laboratory at the Children's Hospital at Westmead. She completed her PhD in 2023 at the University of Sydney, and in 2022 was awarded our Society's Young Investigator Award. Her research focuses on the development application of gene editing tools to treat Nf2-related Schwannomatosis, and her presentation today is entitled Back to Basics, Developing Targeted Base Editing Approaches for NF Single Nucleotide Substitutions. So just bear with me for a minute while I get her presentation up for you.
For this talk, I will specifically focusing on Neurofibromatosis Type 2, or as it's recently been renamed, Nf2-related Schwannomatosis. This is a congenital childhood disease caused by autosomal dominant mutations in Nf2. It has an incident rate about 1 in 30,000. It belongs to a much bigger family of diseases termed the neurofibromatosis, which are grouped together by this predisposition for nerve-associated tumours. For Nf2, this is particularly the development of bilateral vestibular schwannomas, which is what you can see developing in this bottom panel of MRI images. These tumours arise from the Schwann cells surrounding the vestibular nerves, and these vestibular nerves are responsible for hearing and balance, so many of the symptoms are related to that. So we see things such as hearing loss, loss of balance, ringing in the ears, facial weakness, and then there's some more severe phenotypes related to pain as well. We do see other tumours develop in other parts of the central and the peripheral nervous system. What underpins all of these tumours is the development in usually late teen to early adulthood. When we talk about bringing gene therapy to Nf2, I think there's a lot of opportunities there. I already hinted at one in the previous slide, and that's the window for early intervention prior to tumour development or whilst they're still quite small. So this would be more preventative than reactive. There's a plethora of gene editing tools. We live in an exciting era for gene therapy. So we have things such as base editors and prime editors and HIDI. There's also other things such as nonsense suppression therapy that could be applicable in Nf2 as well. The third and not insignificant point is the support of the NF community. There is a range of foundations across the world, such as the Children's Tumour Foundation. More locally as well, we have some philanthropic bodies like the Flicker of Hope that really help to keep an open line of communication between researchers and individuals with NF so that we can understand what sort of technologies they're getting excited by. And gene therapy is one of those. So with that support, it's nice to be able to see that there likely will be clinical translation of these results. There's, of course, a bunch of challenges. Science wouldn't be science without those. Some of those are broadly applied to the gene therapy field, such as delivery to the correct tissue, which, although is very disease-specific, so in NF is Schwann cells. There's a balance of efficiency. Because this is a tumour-linked phenotype, we want to make sure we're hitting as many cells as possible and correcting those. There's availability of preclinical models and access to tissues. These tumours are obviously around the vestibular nerves are quite tricky to get out, and they're often quite rare, so it's hard to get access to those. So how can we develop in-house models that we can use for testing our gene therapies downstream? And that's where this first aim of my project really came in, which was developing different models of Nf2. When we're talking about in vitro models, we just focused on HEC293 cells, primarily because we wanted to get an idea of the readout of efficiencies and whether a base editing or a prime editing approach would be applicable. But to knock it in, we just use a standard CRISPR-Cas9 HDR approach, using a CRISPR-Cas9 and transfecting it alongside a HDR template into HEC293 cells via lipofectamine, and then isolating and screening the genomic DNA from those pooled cells to get an idea of what efficiencies we were looking at before we could create clonal populations harboring these Nf2 mutants. So this is just an exemplar. So this is an Nf2 missense mutation that causes a C to T point mutation within the Nf2 gene. With our pooled population of CRISPR HDR cells, we saw this was around a 16.5% HDR efficiency, which was sufficient for us to be able to pick clonal populations from. And you can see here in this illustrative example, there's 100% knock-in of our C to T point mutation. To complement our human mutant cell line, we also wanted to create an in vivo model or a mouse model of the same variant. When we looked at the sequencing, though, we noticed there were quite a few differences between sort of our native mutant mouse and our human mutant sequence. And so what we ended up doing was humanizing the sequence so that we could have conservation of single guide RNAs across both our mutant mice and our human cell lines. And so that's what we were able to do in this creation of this model. So we understood what was going on at the genetic level, but we are currently doing an aged phenotyping study to understand what's phenotypically may be different with our mutant mouse. To our knowledge, this is the first Nf2 patient mutation mouse line. There are other Nf1 patient mutations for Nf1, but not for Nf2. There are conditional and whole body Nf2 knockout models, and we know those show an increased rate of nerve-associated tumor development. So that's what you can see here from another group with a conditional Nf2 knockout model. This is at 15 months of age, and you can see on the right panel, you can see an increased size of the dorsal root ganglion within the spinal cord that was linked to tumor development. So we're just doing an age study to understand if we are also seeing a same functional effect of our Nf2 mutant. But jumping back to the gene therapy design and validation, this is just a pipeline for our Nf2 mutant. So designing a strategy that's applicable based on PAM site availability and orientation of our mutation that we can then transfect or inject into our models, and then look at the downstream DNA and RNA, and also some more functional measurements of rescue, so looking at tumor burden. I won't labour the point too much on this one. I think many of us are familiar with the base editing technology, but just noting we are using David Liu's SB-Cas9 ABE-Max system for our approach, and that does use a Cas9 nickase variation where you nick the strand opposite to the deamination that you've just currently done to help push the cell's own mismatch repair to repair the mutation rather than retain it. So we co-transfected SB-Cas9 ABE-Max and our Nf2 targeting single guide RNA expression plasmid into our mutant cell line, and then looked at a range of outcomes, including rates of repair of the Nf2 mutation, off-target effects inclusive of bystander effects. So if you look at this single guide RNA sequence on the bottom, you can see the editing window here, and there is a thymine just outside of that. So we wanted to understand if there was any editing going on there, as well as editing of the wild type allele. So looking at some illustrative examples here, you can just see our Nf2 mutant along the top. And with our base editor, even just looking at a Sanger sequencing level, we were seeing quite a high rate of repair, where we can see the vast majority of cells have been changed from a T back to a C at that particular locus. Looking at quantification of that, though, so using targeted amplicon sequencing, we see about an 80% in this particular example, and quantifying that over a couple of different cells. You can see with selection, we're seeing about 70% to 80% gene correction at the 169 locus, noting that Nf2 single guide RNA expression plasmid has puromycin cassette, so we can select for cells that have specifically taken up the plasmid, and then without selection, it was closer to 60%. You would note that we did see some slight bystander effects there. In this example, it's 10%. And then when we look at a range of different cells again, we can see with selection, it was around 10%, and without, it was around 2 to 3%. This doesn't create a missense mutation, which most of the pathogenic Nf2 mutants are, but it does have a small change in the amino acid sequence. We're unsure what this means for the protein, and we are looking at berlin, which is the protein product of Nf2, as well as some of the other proteins in the pathway to understand functionally what this bystander may affect, but we're yet to have those results. But we do have a lead candidate, and we are quite happy with a 70 to 80% gene correction. So we're looking to adapt the system for in vivo delivery. We do note that between in vivo and in vitro, there can be quite big disparities. So it's important to look, even if we slightly change this later on. But adapting to an in vivo-based delivery system, we are going to use an AAV-based system. This particular one we're using, because we're looking at Schwann cells, was devised by our collaborators and the team at the neighbouring institute, the Children's Medical Research Institute. which specializes in a lot of AAV work. And so this was an AAV barcoded human Schwann cell testing kit, whereby each capsid has a unique barcode within their genome. And once they're transduced, and in this case, it was human Schwann cells from either skin or sciatic nerve, as well as fibroblasts. You're able to look at both a DNA and an RNA level to see what capsids show a particular tropism for Schwann cells. And so this is some of the work that came from the mouse studies, where it's primarily done in humans, which is great for clinical translation of this later on. They did do some work in mouse work, which is important for our studies. This was a sciatic nerve crush model and was injected locally into the nerve, whereas we would probably be more focusing on systemic approaches. But you can see here, these are the three candidates. AAV-DJ is something that was designed originally for liver, but there were two new capsids that came out of this study that showed an increased tropism for Schwann cells, and you can see that with the expression of GFP. Interestingly, there were slight differences in the type of or areas of those Schwann cells that it affected, and also some of the types, so myelinating and non-myelinating. But we will be combining these AAVs with our base editing technology. Of course, AAVs, we love them, but they are small. So based on the size constraints with a base editor, particularly with SP-Cas9, needs to be split across two genomes. So we'll be using a dual AAV delivery system with an intine-based system to it, where we split the Cas9 across two as well, and then package the single guide RNA and the base editors into different genomes. We're going to be driving this with either a CAG, ubiquitous CAG promoter, or a Schwann cell targeting promoter, the human CROX20 promoter, and these studies are currently underway. So whilst this project is slightly on the preliminary side, there are some exciting results, and we're really excited to move on with them. We have devised a pipeline to develop NF-CRISPR patient cell lines in-house with about a 3 to 4 month turnaround. We're using this for other NF2 mutations and NF1 mutations. We're the first group, at least to our knowledge, that has developed a mouse model harboring an NF2 patient mutation, and we're going to be using this for a range of therapeutic interventions. With our lead candidate so far, we have shown a high efficiency of repair with limited indels and bystanders, with a little bit more characterization to go on there, and with future work focusing on adapting this to a Schwann cell targeting AAV vector using a dual intine-based approach. But of course, it's a lot of work. Science is that. And with that, I'd like to acknowledge my team. So from the Bioengineering Molecular Medicine group, and then also from our collaborators at the Gene Therapy Research Unit, as well as the important funding bodies that have helped support this work. And with that, thank you for listening.
Thank you very much, Alex, for that lovely presentation. Do we have any questions in the Q&A? Also, just apologies for the sort of false start, but hopefully everything's looking okay now. But if you do run into any problems, just please pop it in the chat function.
We don't have any questions yet, so and everyone that's listening, feel free to put any questions you have for Alex in the chat. Alex, I'll give it a start. I know you said this is early research, but why did you choose this base editing approach versus other gene editing approach for this particular condition?
So in NF, or specifically in NF2, I suppose, there is quite a mutational, but like quite a range of different mutations we see in it. About 35% of them are point mutations. And while some of the more broader strategies, so some of the work with HIDI or some of the prime editors may be a little less bespoke, because of the high efficiencies that's probably going to be required with it, because of the tumor like phenotype, we're going to probably have, that's going to be finding a balance of something where you is not so bespoke, but also high efficiency, that meant that we were more drawn to base editors at this stage. But I think as the technology all advances the field, there's probably going to be a forerunner that comes out. That even if we make one difference by sort of working up a vector or working up a specific technology, that will mean that, you know, something will have some clinical translation.
Yes. And is there any particular hotspots?
Nope, unfortunately not. We see it pretty much in all the exons. NF2 is not the biggest gene. You can fit the cDNA into a single vector, but there's issues of balancing because it's cancer-like, balancing overexpression. That would be an issue there. So that's why sticking with maybe more of a precision editing will be better off in this situation.
So Alex, can I ask a question? You just touched on it briefly in terms of efficiency. Do you have a sense of, and it could be a difficult question to answer until you actually do it, but do you have a sense of how many cells you might need to correct in your model?
That's a big question. And I think in NF2, partly because of the problem, because even though it is an autosomal dominant disease, because it's really actually linked to haploinsufficiency when you have that second hit inactivation, we know the tumors develop after that. So it's an issue of, well, if you have enough expression of that gene in there, will you have less tumor development? I don't think I'm comfortable in saying a number. I think in this particular disease, it's harder than some others. But I think whatever it is, it's going to be probably higher than what we've seen in other gene therapy approaches for the liver and the retina.
Still don't have any questions, so I'll have one more as well. Alex, you mentioned systemic approach for the delivery. That's obviously in the future, but then for your in vivo model as well. So what do you have to consider when looking at the safety of targeting effect for a systemic approach for this type of therapy?
Yeah, so because most of the tumors are in the central and peripheral nervous system, it's probably going to be balance of, I think it's probably going to be more intrathecal that it ends up being. We know that's something they do with AAV-based gene therapies that are already in clinic, so there is a pathway there. If you did end up going more systemic, it's probably more important for some of the other NF diseases, so NF1, which affects a lot more tissues. It's a little bit more specific in NF2. But of course, as soon as you go systemic, you've got to think about all your off-target organs. The liver loves to soak up anything you put into the system. So of course, the safety concerns with that would be pretty major.
We do have a question from the chat. So they're asking if you looked at off-target editing of the adenine base, editor.
I would have loved to present that data. It is currently in the process of being sequenced. So we do have there's two particular off-target sites with this base editor that we're more concerned about. It does take at least 3 mismatches to be tolerated. But of course, we have a range of other ones that we're also going to be looking at. And then eventually down the line, when we're more set specifically on definitely using AB Max, or maybe trying something that maybe could minimize some of the bystander effects, then we would look at doing some more whole genome sequencing based work as well.
That's great. We don't have any other questions.
Okay, well, in that case, I might introduce our next speaker. So thank you again, Alex, for your lovely presentation. So our next speaker is Professor James Hudson. So James completed his PhD in biotechnology at the University of Queensland and went on to do a postdoc in Göttingen in Germany. So James now runs a lab at QIMR Bergenhofer, bringing together the disciplines of engineering and cell biology to develop human cardiac organoid screening platforms. He's also generating a comprehensive map of the molecular processes governing cardiac function and looking to translate key therapeutic candidates for cardiomyopathies and inflammatory disease-driven dysfunction. James is an inaugural recipient of the Snow Medical Fellowship in 2021, and today James will be presenting his work entitled A Transient Modified mRNA Encoding MYC and Cyclin T1 Induces Cardiac Regeneration and Improves Cardiac Function After Myocardial Injury. So I'm going to bring up his presentation now, and I'll try and do everything all at once.
My name is James Hudson, and I'd like to thank the Australian Gene and Cell Therapy Society for inviting me to present some of our work today. Today I'm going to be focused mainly around our work in collaboration with the University of Cambridge and AstraZeneca on mixed cyclin T mRNA. So firstly, just to give you an overview, heart regeneration would really transform cardiac care. So there's over 50 million patients worldwide with heart failure. This is projected to increase substantially over the next 3 decades due to our lifestyle factors and other community risks. And also, it's the most costly burden to our healthcare system with no cure. What typically happens in someone that might have an infarct, myocardial infarction, or a other insult, which can be caused by other things such as genetic diseases or viral infections, is you lose a proportion of your cardiomyocytes, and this can be up to 1 billion of the cardiomyocytes in the heart. Your heart has about 4 billion cardiomyocytes. And because these don't regenerate, what happens is the remaining cardiomyocytes undergo a process called hypertrophy, so they get bigger to take that contractile load. There's also substantial fibrosis to hold the heart wall together. And together, these things result in an even worsening in pathology of the heart and heart function, leading to heart failure. One of the seminal studies performed by Enzo Perello, who's an Australian scientist at MCRI, was that regeneration does occur in mammals, but it's just rapidly lost in the postnatal window after birth. And so what he showed is that if you give a myocardial infarction to a 1-day-old mouse, the heart can fully regenerate the lost cardiomyocytes, and it can push out the scar tissue, so you have no remaining scar tissue. And this is a section through the midsection of the heart. What you can see here is already by day 7, you start to get more scarring of the heart, and this progressively worsens as the mammal matures to adult. And this has also been now recapitulated in large animals such as pigs and shown that this also occurs. And there's also some case studies in humans suggesting this might be the case too. Now, rather than a stem cell compartment, what he showed was regeneration occurs via cardiomyocyte proliferation. To do this, he did a nice lineage tracing study whereby you do a pulse chase analysis. And if the cardiomyocytes are LAC-C positive, they're essentially renewed by pre-existing cardiomyocytes. But if they're not positive, then they're renewed by progenitor cells. This labeling isn't fully efficient. You get around 70%. But as you can see here, in both the sham surgery and after myocardial infarction, this percentage remains the same, indicating that the replenished cardiomyocytes come from cardiomyocyte proliferation rather than any stem cell compartment. This has also been shown quite comprehensively in various studies showing that this proliferation really reduces after birth. And so instead of now getting number expansion of cardiomyocytes, you start to get this multinucleation phase and polyploidy as you progress to the adult. And this is in mice. So mice are around over 90% of the cardiomyocytes have 2 nuclei. And this varies across the animal kingdom. So in humans, actually only around 25%, and a lot of the cells are polyploid. Similar tracing studies have been done in humans, where there was a lot of atomic testing done earlier in the 20th century. And the incorporation of C14 carbon into cellular matter can be traced in humans over time. And essentially what researchers did was performed analysis of heart sections from humans at all different ages. And they were able to calculate that the turnover of cardiomyocytes in the human heart is only less than 1% per year. So really, there's very little repair that can happen in adult humans. We wanted to dissect what happens and what occurs during this postnatal window, and particularly so that we can try and figure out mechanisms that we can reawaken this regeneration process. And so Greg Quaife-Ryan in our lab, he fact sorted different cardiomyocyte cell populations And we had sham operations at both day 1, postnatal, and then took these samples 3 days after that sort of sham operation. He also performed myocardial infarction and took samples 3 days after that, and did the same thing on adult mice at day 56. What you can see here is this is a PCA plot of RNA sequencing, where each dot represents a different sample. And basically on this plot, the further apart the dots are, the more distinct their phenotype. What you can see here is that fibroblasts, as the fibroblast and the heart matures and progresses into an adult, these change substantially in their phenotype. However, after injury, these really revert back to a more embryonic type state. You could see a similar thing for the endothelial cells, although not quite to the same extent. A similar thing for the leukocytes. However, the cardiomyocytes fail to revert much at all back to their phenotype in the early postnatal window. And this reversion is essentially dictated by cell cycle transcripts. And so it essentially indicates that these other cell types can go into a proliferative cycle to repair the damage. However, cardiomyocytes cannot. And what underpins this massive change from fetal to adulthood in the cardiomyocytes is really underpinned by lots of metabolic changes, including oxidative phosphorylation. And so what we did was we used this data set to try and identify pathways that might be activated in a neonatal cardiomyocyte that aren't in an adult that we can utilize to drive cardiomyocyte proliferation. When we looked at what was most substantially regulated was really the Wnt signaling pathway and sort of transcripts that are dictated by beta-catenin signaling. And so using a special antibody that only marks activated beta-catenin, what we showed is that at P1, you see cardiomyocytes are positive in their nuclei for beta-catenin, and this progressively drops during development to where there's really no active beta-catenin anymore. We also see a drop in the beta-catenin transcript, even though it's regulated post-translationally for its activation, and also its cofactor. So it binds to TCF7L2 is the name of the gene, TCF4 is the name of the protein, and it binds this protein to activate genes, and this also drops off. We wanted to assess what transcriptional targets of beta-catenin were, and so we performed chip sequencing, and we did this on its cofactor that sits bound to the DNA. And we also did 2 different histone marks that act for active transcription. You can see that this chip-seq was really enriched for TCF-LEF. So this is consensus sequence of TCF where beta-catenin binds. And this also overlapped a lot with enhancer or promoter peaks, which are marked by these 2 different histone marks. And so we really think that beta-catenin is controlling a lot of these genes and transcription of these genes at the genome level. We then perform RNA sequencing on Kaya 99021 treated cardiomyocytes. And this is basically to activate the Wnt beta-catenin signaling pathway. It's a GSK3 inhibitor. And what you can see here is that this activated a lot of different genes that also overlaps with these enhancer peaks, all the promoter peaks, and a TCF7L2 chip-seq event. And these genes are mostly characterized by cell division, DNA repair, and telomere maintenance, all genes heavily involved in regulation of cell cycle. Interestingly, there's what we saw in the down-regulated genes is a response to hypoxia and negative feedback to sort of repress the Wnt beta-catenin cycle. And so we wanted to assess whether beta-catenin was involved in proliferation in neonatal cardiomyocytes. In order to do this, we treated neonatal mice with injections of IWR1, which is a axon 2 stabilizer and inhibits beta-catenin activation. You can see that this reduces proliferation in the hearts in vivo. And when we overexpress a constitutively active form of beta-catenin, which we delivered using an AAV6 vector, we could boost proliferation in the neonatal window and also cardiomyocyte number. And so this strongly suggests that the Wnt beta-catenin pathway is heavily involved in cardiomyocyte proliferation and potentially can drive regeneration. We did further experiments in adults, and so what we did here was in adult mice, we delivered constitutively active beta-catenin. We also delivered constitutively active YAP S127A, and this was already, we use this as a positive control because it's been shown in the literature to activate proliferation. We traced proliferation using BRDU, and we also performed echocardiography over time to determine if there was any functional benefit. Very interestingly, we found that both of these protected the hearts to the same degree. So we really got this improvement in cardiac performance marked by ejection fraction. That's essentially the amount of blood pumped out with each beat, which really drops in after a heart attack. And we can see this enhanced performance. And it's similar for both these factors. So we got quite excited. However, what we found was that only YAP activated proliferation, and to be honest, quite a minor degree of proliferation. And beta-catenin didn't activate proliferation at all. So we think this is more a protective effect. We performed RNA sequencing on purified cardiomyocytes from those adult hearts and looked at the sort of regeneration network marked by the CHIP sequencing and in vivo RNA sequencing results. And what we found was that in the adult heart, beta-catenin really gets skewed to these metabolic factors and activates metabolic genes rather than the sort of transcriptional response driving proliferation. And so it really switches and redirects the genes that it's targeted to. Now, this is potentially more of a transcriptional regulation rather than chromatin closing, because when we look at our data sets on ATAC-seq of the hearts, we actually find that none of these regions are closed off during development. And so it's really the way that the cells control transcription rather than a epigenetic shutdown and silencing. And so what we hypothesized could be happening is that this metabolism and this oxidative phosphorylation really drives the cell cycle arrest process. And using our human cardiac organoid model, we actually showed that this is the case, so that switching metabolism to oxidative phosphorylation shuts down the cell cycle, no longer enables proliferation, and also shuts down Wnt beta-catenin driven proliferation in that model as well. You can really see this highlighted here. So this is neonatal cardiomyocyte RNA sequencing data, and this is overlaid to all of the pathways in the KEGG database. And so you see all the different colors are all the different metabolic pathways. And in black, all of those that are high in neonatal cardiomyocytes versus adults. And you can see there's a lot of branching between different processes. And not really that much activity down towards the TCA cycle. What you see in the adult cardiomyocytes is there's a lot more interaction between the pathways, and these really funnel into the TCA cycle. And so what we think is happening is the cardiomyocytes are funneling their metabolism into the TCA cycle for energy. Cardiomyocytes are one of the most energy intensive cells in the body. You only have a couple of seconds of ATP unless they're actively producing ATP and energy. And so around at the same time, Kathy Wilson and Megan Bywater at the University of Cambridge identified that a MYC complex with RAS rewires metabolism. And they showed this in vivo in the mouse, and that using RNA sequencing, it really reverts this metabolic pathway back out to this branching and no longer funneling everything into the TCA cycle. And importantly, MYC is a really critical beta-catenin target gene that's activated in cancers, but also in the neonatal cardiomyocytes with Wnt activation. Kathy and Megan showed that numerous organs would activate proliferation by just driving MYC, as you can see here. However, the heart and the brain, which are key organs that have no regenerative capacity, activate this process. And what they showed that you also need cyclin T1 to drive this process. And they really got profound levels of proliferation with this complex being activated. And we even worked together with James Ray and Rob Pardon at UQ and took 3D TEMs and showed, you know, for one of the first times, a TEM of a bionucleated cardiomyocyte undergoing cytokinesis. And so we really think this mixed cyclin T has potential as a regenerative therapeutic. However, to use this as a gene therapy is really challenging. We need to localize it to the heart, and that's because it's a potent oncogene. It needs to be localized to the border zone, as whole heart expression can result in sudden cardiac death. And it needs to be transient and non-integrating into the genome, as this would pose a significant oncogenic risk, and ongoing proliferation would also be detrimental. And so we started working on modified mRNA to do this, and this was well before the COVID pandemic, where now nearly everyone is using modified mRNA for different therapeutics. And so for this project, Alexandra Boykova over at Cambridge with Kathy and Greg Quaff Ryan in our lab developed this project where they're delivering modified mRNA and various other genetic forms of transient delivery to sort of reveal that we could utilize this as a therapeutic pathway following myocardial infarction. I don't have time to go through all the details of this, but you can read most of it in our bioarchives manuscript. As you can see here, injection into the heart wall, and you can see here with TD tomato Cree lineage tracing that you really see localized delivery of the modified mRNA around the injection site. And these are sections through a heart where you see once you get down into other sections away from this injection site, you no longer get expression. So the delivery is really localized to a specific region where you inject the modified mRNA. The other thing we showed using bioluminescence and IVUS imaging of mice is that it's extremely transient. We weren't expecting this. We actually, based on literature, thought that the peak would be out around 2 or 3 days, but actually it's really fast. Within 8 hours, we're getting maximal sort of expression. And in fact, I'll show you some data later where it's even earlier than this. And so after this peak, it rapidly declines over time. And so this is perfect for a regenerative therapeutic where you're delivering MYC, where you only want a transient response. As you could see on that IVUS, we looked at delivery to other organs, and you get very little delivery to other organs. So, you know, less than 0.5% to the spleen and liver, which really overlays that IVUS image you just saw on the previous slide. And so most of it is going into the heart. And because this is transient and not permanent delivery, this poses less of a risk. One thing, though, that is important is that we do get a proper proliferative response with such a transient delivery of mixed cyclin T. And in order to investigate this, we delivered the modified mRNA of mixed cyclin T, which is fused together by a T2A in a single construct. And we used human pluripotent stem cell derived cardiomyocytes to do these experiments. We delivered mixed cyclin T, and then we did proteomics at different time points to investigate activation of proliferation and various other pathways. You can see that both MYC and cyclin T are really fast upregulated during this process. Already by 1 hour, we have really substantial expression, and cyclin T is prolonged for a bit longer, but MYC is abruptly shut down, potentially through negative feedback mechanisms. As you can see at these early time points, there's very little proteins upregulated, and this response really takes place over time. So even though MYC is rapidly shut down, this cell cycle network really progresses over time, over 48 hours, to activate a whole protein network. Now, this induces a proliferative response. So we get around 25% additional activation of Ki67, and these are proliferative 2D cardiomyocytes, so they're pretty immature. And we get a reduction in cell size, which is pretty standard with cardiomyocytes. And we get a boost in cardiomyocyte number. Now, these changes in cell number aren't massive, as you'd expect in the cancer field. But remember, we only need around 25% of the cardiomyocytes to divide once in the heart to fully regenerate the lost cardiomyocytes. So we're not looking for huge changes in cell number with these sort of experiments. This activation of proliferation over time really coincided with activation of MYC target genes, mTOR signaling, and cell cycle targets that are really critical for cell cycle progression. We also looked at its injection in vivo. So this was in a myocardial infarction model in adult mice. We injected mixed cyclin T, and we looked very shortly after at the hearts. You can see already by 24 hours, similar to the bioluminescence experiments I showed you, you already get a sustained delivery of GFP, because GFP hangs around for a longer period of time. But we really saw the MYC and cyclin T drop off by this time point. So this is just after an hour where you really see substantial upregulation of these pathways. So sort of when you deliver a modified RNA, the response and the production of that protein is very, very fast, and it's very, very transient, depending on what you deliver. We also showed that in vivo, in that same paradigm, MI with injection around the border zone, we get elevated EDU cardiomyocytes, we get pH3 positive cardiomyocytes in mitosis, and we quantified that for cardiomyocytes here, but also quantified just pH3 per section to show that it wasn't really inducing proliferation in the non-myocyte population. And with this, we also see after a longer time, about 5 days, an increase in cardiomyocyte number, which is really important for regenerative therapeutics, where you don't want to just increase multinucleation. So we set up another experimental paradigm to assess whether transient mixed cyclin T can improve heart function in vivo. Similar sort of experimental paradigm where we do a baseline echo, and then we do injection of mixed cyclin T around the border zone together with an MI, and then follow up at different time points out to 28 days to look at function. What we see here is a improvement in systolic heart function. So this is ejection fraction, this is the amount of blood pumped. And you also see that when you look at percent decline from baseline, this is substantially improved in the mixed cyclin T animals. So this is really inducing a proliferative response that results in sustained cardiomyocytes that improves heart function. So it's very exciting. We next wanted to show that this wasn't just cardiomyocyte protection. And in order to do this, what we did was we can't really open the chest back up at a decent time point later and then inject the modified mRNA. And so we did this using a genetic approach. So we deliver AAV cyclin T 4 weeks before surgery. And then what we do is we do an myocardial infarction where we actually deliver a modified mRNA encoding a CRE. This enables us to activate MYC by giving transient tamoxifen, so we can sort of dictate what time we would activate MYC. And so this was done 7 days after myocardial infarction in this experiment. And then we assessed the functional improvement afterwards. And what you can see here is that both mice with the control or the MYC activation result in a similar decline by 3 days. And then once we reactivate MYC, we get this boost in heart function, which is sustained over a long period of time when we reactivate MYC. And this is also shown here in the delta heart function. And so in summary of this, we think that MYC cyclin T using modified mRNA is a very powerful gene therapy to try and improve heart function after heart attack or other diseases where you might lose cardiomyocytes. So in summary, mammals have a transient window where they can regenerate their hearts endogenously, and this is rapidly lost. Reactivation of proliferation really requires metabolic rewiring, and simple overexpression of the developmental pathways is not sufficient. MYC cyclin T can rewire metabolism, and transient expression using modified mRNA drives proliferation and also improves heart function. And so next, the key to translation of this will really be targeted delivery strategies, where in a human we don't have to directly inject the modified mRNA, but we can use particles or targeting antibodies or peptides to deliver that to the heart, and also enable multiple doses to be given. With that, I'd really like to thank Kathy and her lab, together with Alexandra, on this project. This is a really, long-term collaborative project that's been very exciting and successful. Also, Greg, he did a lot of the early regenerative work, and later the work on MYC cyclin T, together with Kathy's lab. Chris and Harley did a lot of the 2D cardiomyocyte experiments, looking at both activation of proliferation, but then also the proteomics. Enzo Perello, a long-term collaboration on cardiac regeneration, and later development of the MYC cyclin T sort of approaches for mRNA. And then Jingdong at AstraZeneca for collaborating with us on this project and providing insights and modified mRNA. So with that, I'd like to thank you all for listening, and I'm happy to take any questions.
Thank you so much, James, for that really lovely presentation. Does anyone have any questions?
Again, as we said, if you've got any questions, please put them on the Q&A down here at the bottom. A great talk, James. Thanks for that. As you were talking, I was thinking of questions, but you seem to be answering them on the next few slides as soon as I have a question. So one bit technical question on the tamoxifen model, how long is MYC activated for? Is it similar to, does it mimic the same as the mRNA in terms of how long it's active?
Yeah, it's quite transient. It's given with just injection to the animal only for a day. So it probably is a little bit longer, but we don't fully know the kinetics because, as you saw with the mRNA, it shuts down extremely rapidly. We would need to do a bit more profiling of that. But yeah, it is very transient as well.
But it seems, as you mentioned, there's a self-regulating mechanism helping.
Yeah, for the MYC,
as a therapy, potentially, quite safe.
Yeah, even, something like the luciferase rapidly declines. So I think, there's not necessarily inbuilt negative feedback mechanisms for that. And so you could assume that that's very similar to the profile you'd see without any negative feedback mechanism. And then basically you're probably going anything below that because nearly all of the other proteins that are endogenous will have some form of regulation.
We'll give it a few more minutes in case people are feeling a bit shy about it. I'll keep asking questions. How do you see it coming into the clinic for patients, and what will be that ideal window for treating patients?
Yeah, that's a good question. I think there's actually two separate answers to your question. I think the window where patients will probably first get it will be more severe heart failure patients where a new therapeutic will be tested. Where they've got limited options and things like that. However, the time where it would be most effective is actually really early after myocardial infarction, because then you'd prevent the heart remodeling. And this is a bit of an issue with like lots of different heart failure strategies, because the earlier you get in, the potential more benefit you can have, but it's also challenging to try and do a experimental new drug at that time point as well. So you're sort of caught between this sort of trying to get in early, but also trying to make sure you're only testing this on really sick patients initially. Yeah.
We do have a question from the chat. So Carlos Javier Almaciega Diaz is asking, a very nice presentation. Although you are using a transient approach, it seems that the metabolic effects stay for a long time. Do you anticipate any side effects of this long-term effects on the metabolic changes?
Yeah, I think the metabolic effects will be quite transient as well. So when we use the original genetic models in the RNA-seq data I showed you, that's over a longer time period. I think the metabolic changes itself will be quite transient with that approach, and that's sort of what we see. If you prolong a metabolic insult that decreases oxphos, you'll start to lose contractile function, and as you saw from the in vivo data, that we don't really see a loss in function if we just give a transient sort of dose. When we give in the genetic model, if you activate MYC and cyclin T for a longer period of time in the whole heart, that can be quite toxic, and you get deregulation of a lot of ion channels and things like that too. And so it's really important to have the localized and transient delivery so that you don't get the prolonged metabolic and sarcomeric changes and things. Yeah.
And how much of those changes, because you mentioned the TCA cycle early on, does that play a big role with these as well?
We're doing a lot of mechanistic work right now on how contraction and metabolism controls regeneration. There's a lot of literature too. It's been well established that increased oxidative phosphorylation and TCA cycle activity will drive cell cycle arrest in cardiomyocytes. However, the exact mechanism of that isn't really known. It's known as a DNA damage response, but it's not known sort of how that happens. It's hypothesized this could be oxidative stress and things like that. But if there was full-blown oxidative stress over your lifespan, your heart would likely severely deteriorate in function with a lot of mutations. So this is an active area of research that's really important for the field, I think.
So I might just ask a question. You sort of touched on it in the end with sort of delivery systems. Is that something that you're working on as well? How would you, deliver?
Yeah, we're doing a little bit of work on that because it's a very important area. But we're wary of doing too much. We've got some unique strategies that we want to try, and we'll see how they work. A lot of people have tried nanoparticles and things like that, and it is quite challenging and quite a difficult thing to achieve. So I think we'll see what happens. Since mRNA sort of became a major vaccination target, and companies like Moderna want to also get their modified mRNA products out into other diseases, this area of research has really expanded. And so hopefully someone develops strategies that work really effectively.
Yeah, obviously the liver's, going to always be a, prime target. So
Yeah, yeah,
the liver is
yeah.
a challenge. Definitely.
There's some really there's some unique sort of perfusion methodologies that people use to deliver AAVs and things, and those could help sort of de-target the liver in combination with some sort of heart targeting at the same time. And so there are those sort of strategies that can be pursued as well. Obviously, they're a bit more difficult to administer and require, a lot more equipment and expertise by the cardiologists, etc. And so that would also be detrimental to trying to get it out into a broad range of patients too.
I
Don't
can't.
seem to have any more questions in the chat. And I guess, yeah, you've answered all of mine as well.
So maybe if there's no more questions, I'd just really like to thank our two speakers, so Alexandra and James, for sharing their expertise, and also thank everyone for joining the session of the Around the World webinar series. So thank you very much. It's been a pleasure hosting it, and we look forward to seeing you at the next one. Okay, thank you, everyone.
Thank you.
Thanks, everyone. See you later.
Thanks.